Okay, so this part's going to be a little bit harder. Here we're talking about actually physically how are we copying this DNA and building the new strand. So there's an enzyme here called DNA polymerase, and the function of this enzyme is to build the new strand of DNA one nucleotide at a time. So if we look at the blue strand here, this is the parental strand, and the gray strand is the new daughter strand. DNA polymerase is the enzyme that's actually adding the nucleotides onto the daughter strand. There's another enzyme to learn here called DNA ligase. DNA ligase is basically the gluing enzyme. So this is the enzyme that's going to connect the different strands together. So we'll learn more about that in just a minute. So this process doesn't just happen with one bubble. There's actually many bubbles that form simultaneously. In other words, this origin of replication, or where we're starting the replication, there's actually many, many origins of replication in one DNA molecule at the same time. And the reason for this is just for speed. If there's more origins of replication, if there's more bubbles, the DNA molecule can be copied faster. And the faster the DNA can be copied, the faster the cell can divide. So we want this process to be fairly quick. So each of these bubbles are going to work the same way. We're going to start at one point. We're going to open up these DNA strands by separating the two parental strands. And then we're going to build in the new gray strand here, which will be complementary to the parental strand. When we finish this process, we'll have two daughter DNA molecules that are identical to the original parent DNA molecule. Okay, now this is probably the hardest part, but take a look. This is some organic chemistry notation. So in each of these deoxyribose sugars, we have these 5 carbons that are labeled with prime. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And if you look at the 3 prime and the 5 prime, you'll notice that on the left strand, 3 is on the bottom and 5 is on the top. But on the right strand, 3 is on the top and 5 is on the bottom. So what does this mean? Well, it means that these two DNA strands are facing in opposite directions. And that's actually going to be really important for DNA polymerase. So when we look at the DNA molecule, whichever side, the 3 prime or the 5 prime, whichever side is facing up, well, right here, that would be 5. So then this end would be called the 5 prime end. And since the 3 is facing down, this end of the molecule is called the 3 prime end. If we look at the other strand, it's exactly the opposite. The 3 prime carbon of the deoxyribose is facing up. Therefore, this would be the 3 prime end. And the 5 prime carbon is facing down. So down here, this would be the 5 prime end. So when we look at a DNA molecule, one strand is always going 5 to 3, and the other strand is always going 3 to 5. They're opposite. If we look at this molecule here, this top strand is going 5 to 3, and this bottom strand is going 3 to 5. Now, why does this matter? Well, this matters because DNA polymerase, which is this enzyme that's going to build the new strand, just like we saw here. Remember, we're building on this new strand by adding in these new nucleotides. DNA polymerase can only read the parental strand going from the 3 prime to the 5 prime direction. So when DNA polymerase is reading this strand, everything is good because DNA polymerase can move from 3 towards 5, and it can build this strand here. However, the other strand, which is being unzipped 5 to 3, there's a problem.